Bethany has an academic background, fascinatingly, in English and physics, and she's just coming to the end of her PhD in quantum biology at the University of KwaZulu-Natal. Now, Bethany also works as part of our team in science communications, and she's recently undertaken some theoretical research investigating magnetic fields across the galaxy. Bethany's going to talk to us about the overview of magnetic fields in biology. Bethany, over to you.
Thanks so much, Jeffrey, and I'm very, very glad to be able to be part of this very exciting seminar. So we've all heard about electric fields in biology, and I'll be giving a very brief overview of magnetic fields in biology before Prof. Wendy Beane gives us a more detailed idea of how magnetic fields can have a very real, measurable effect on biological organisms. We've got quite a strict time, so I'm not going to go into any detail about the physics of magnetic fields, but more give you some idea of how they might play a role in living systems, both from the point of view of space travel, but also importantly, life and health on Earth. So at the risk of simplification, a magnetic field might be described as the space or region in which a magnetic force is experienced. Magnetism was first discovered as a property of certain materials, such as lodestones, which are composed of naturally occurring magnetite, which exerted an attractive force on iron, for instance. Historical use of magnetism has included navigation, but also actually medicine, used to draw arrowheads out of wounds, as described in ancient Indian medical text. And magnetite also occurs in biological materials. Michael gave us a great overview of electric fields in biology. And while electricity and magnetism used to be thought of as separate phenomena, scientists such as Oersted and Faraday and Maxwell were instrumental in demonstrating experimentally and theoretically that they were related phenomena, which led to electromagnetism and understanding of light as electromagnetic radiation. So it was further developed in the context of quantum theory and particle physics, which the details of which are beyond my time constraints to go into. So magnetism in a material can be understood from the electronic structure of the material and the arrangement of magnetic moments, which instead of being aligned randomly, align in such a way as to sum up to an overall magnetic field. And a magnetic field can also be induced by an electric current or the movement of charge in a material. So why are magnetic fields important in the context of biology and space travel? Well, because life evolved in a magnetic field, that of the Earth. Currents created by the Earth's moving liquid metallic core give the Earth its magnetic field. And this field ranges in strength from around 30 microtesla at the equator to around 70 microtesla at the poles. There are also stronger and weaker patches, such as the South Atlantic anomaly, which is even weaker than the equator. So how does this field play a role in life and biology? Well, we've heard from Afshan about cosmic radiation, and we know the magnetic field is essential for protection from harmful cosmic radiation. But does the Earth's magnetic field play a more direct role in biological systems? Are certain features in living organisms perhaps coupled to the Earth's magnetic field? So living organisms are evolved in the specific context of the Earth's magnetic field, then it is useful to know how this compares to other magnetic fields that might be encountered, for instance, in the context of space travel. For example, in our solar system, the magnetic fields vary from smaller than the Earth's field, even zero magnetic field on some of the planets, to stronger than the Earth's magnetic field. Should longer term settlement take place on Mars, for instance, then we probably need to understand the role that magnetic fields exert, not only for shorter periods of time, but also in the context of reproduction. And this seems especially important because a lot of the emerging evidence seems to show that magnetic fields have an effect on stem cells. So it's impossible to talk about magnetic field effects in biology without at least introducing the idea of spin. In the history of quantum mechanics, spin makes its first appearance in the behavior of atoms in a magnetic field in the Stern-Gerlach experiment. And Pauli's famous exclusion principle used spin to ensure that two electrons don't occupy the same quantum state. Spin is defined as intrinsic angular momentum, which is where the term spin comes from. But intrinsic meaning it is something that the matter has rather than something that it does. And there's still quite a lot of debate as to what exactly spin is. So to avoid too much confusion, I think it's probably easiest to think of it as the property of matter that describes how a particle, such as an electron, behaves in a magnetic field, in the same way that we are quite used to thinking about charge as the property of matter that is acted on by an electric field and mass as the property of matter that is acted on by a gravitational field. So spin has already played an important role in medical technologies. It gave us magnetic resonance imaging, which uses strong magnetic fields and spin relaxation as an imaging tool. The fundamental science that gave us the MRI is nuclear magnetic resonance, but NMR techniques were only developed into the medical context in the 70s. NMR targets nuclear spin and uses strong magnetic fields, much stronger than the naturally occurring fields, such as the Earth's. So what about these weaker fields? So one of the ways in which weak fields might play a role in biology is through their influence on chemical reactions. Spin chemistry is a field of research that was established already in the 1960s to explain how magnetic fields can change the outcome of a chemical reaction by changing the spin states of paired electrons. This is known as the radical pair mechanism and involves the creation of a pair of radicals or electrons whose spins are correlated in one of two possible states. And this correlated spin is then manipulated through magnetic field interactions, and the resulting spin state leads to distinct different chemical outcomes. The role that spin might play in biology has also come under the spotlight because of chirality. Chirality is a description of mirror symmetry or handedness. And chirality is particularly interesting in the biological context because of the fact that biological molecules such as amino acids have a distinct chirality rather than being a mix of both right and left chirality. This is interesting in the context of quantum biology because of the focus on charge transfer in processes such as electron transport chains, mitochondria, and photosynthesis, because it turns out that chiral molecules preferentially transfer electrons with specific spin, causing spin polarized currents. So NMR and MRI look at nuclear spin in strong fields, but what about nuclear spin in weak fields? Magnetic effects associated with electrons are often prioritized in this context, like the radical pair mechanism, because the magnetic moment of electrons is much stronger than nuclear. But there is some research into how nuclear spin correlation could lead to biological outcomes at weak fields, especially in the context of calcium phosphate and how this could translate into calcium ion production and neural activation. So there's actually a surprising amount of experimental evidence that magnetic fields affect the growth of plants. This effect manifests in different ways, including differential growth as well as seed germination. But the progress has been slowed by the fact that these experiments give conflicting evidence and are sometimes unstandardized. In animals, magnetic field effects have mostly been studied in the context of navigation, and there's still some debate as to whether the mechanism behind this is the radical pair mechanism or due to endogenous magnetic materials, such as magnetite. More recently, researchers have begun to investigate the role of magnetic fields in other biological processes, such as stem cell growth in Planaria, which Prof. Beane will be telling us about. There's also been some new research into how magnetic fields affect stem cells and muscle regeneration. And there seems to be growing evidence that magnetic fields affect reactive oxygen species, which are fundamental to biological processes. So while we should be concerned about changing magnetic fields in the context of space travel, this also might lend us some insight into health on Earth. So the Earth's magnetic field varies by latitude from 30 to 70 microtesla. And there are a number of diseases with latitude dependence, so multiple sclerosis, inflammatory bowel disease, infant spasms that are consistent with epileptogenesis, and even COVID-19 show this latitude dependence. What is potentially interesting about these diseases is that they all seem to be inflammatory diseases. And could magnetic field differences contribute to differences in inflammation, with reactive oxygen species modulation being the underlying mechanism at play? Of course, there are other latitude-dependent aspects to take into account, such as light differences and vitamin D. And finally, I've talked about living systems and artificial magnetic fields, but what about artificial living systems and magnetic fields? In one of our previous meetings, Prof. Guy suggested it would be interesting to know how artificial life forms, like the xenobots developed by Prof. Mike Levin and colleagues, would react to different magnetic fields. Xenobots are novel life forms consisting of frog skin and heart cells. While the effects of electric fields, such as membrane potential, will no doubt be important to understand in this context, it might also be interesting to see how magnetic fields different to the Earth's affect these artificial life forms. So in conclusion, very briefly, before Prof. Beane gives us more of the interesting details, it seems that magnetic fields are important in the context of living systems, but consistent experimental data is lacking. And a better understanding of this will contribute to efforts to settle in space, but also more immediately to our understanding of life and health on Earth. Thanks very much.
Betony, thank you very much. Concise and succinct as usual. Very, very good. Well done. And I am absolutely delighted to welcome Wendy, who has literally been online for about 30 seconds, as you said, and Wendy Beane is going to be talking about this. Wendy, welcome to our meeting, and thank you for arriving on absolutely on time. So Wendy is the Associate Professor in the Department of Biological Sciences at Western Michigan University, which is in Kalamazoo. And she spoke as part of our autumn series of online lectures last year. And we're very, very pleased to hear her again. So Wendy's going to be speaking about reactive oxygen species and stem cells in respect to magnetic fields. So Wendy, if you would like to share your slides, we'd be very pleased to receive your presentation.
Thank you for that nice introduction, and thank you to Betony for doing all the hard bits and explaining the technical stuff so I can just play with my pretty pictures, which is the goal of most biologists. So what I really wanted to talk to you about was how radical oxygen species and signaling downstream of it affects stem cells, and more importantly, how we're realizing that we could use magnetic fields to influence our events. So my lab studies stem cells, which are unique and a bit weird for cells because they have an ability to give rise to cells that are just like themselves, as well as cells that are completely different from themselves. And and this means that stem cells are an important driver of tissue growth. And we really want to study tissue growth, and we'll tell you why. So here you have got two pictures of two different organs, a liver on the left and a heart on the right, and both of them have a blue area of tissue growth. However, all growth is not equal. What we have on the left is a tumor, and on the right we have a zebrafish heart, which is able to regenerate new tissue. So we really need to figure out what the mechanisms are between these two, how we can regulate it, and where they differ, so that we can promote it when we want and stop it when we don't. So to do this, my lab uses the Planarian stem cell model system. They are awesome. They run about less than a centimeter long with the species that we have. They're free living, non-parasitic. They can be found in at least a species can be found in pretty much any lake, stream, or riverbed of your choice, as long as you aren't in Antarctica. And we really like them because they have unique features. Apparently, I like studying things that are a bit odd. And they have an unbelievably high percentage of adult stem cells. So we only study the adult. And whereas you or I have about 1% of our bodies comprised of stem cells, the Planarian has anywhere from 25 to 30% of all of their adult cells are stem cells. You can see them here in green. And what this means is that they are a powerhouse model of regeneration. So you can see here a single worm on the left that we have cut out the middle fragment, and these are the fragments, the fragment that resulted from this exact worm. And over several weeks, it is able to completely regenerate that fragment into a whole new worm, even though it's smaller. And even though it didn't start with any brain, it can reproduce all body parts, including a brand new brain. So what we're interested is obviously how it's able to do that. That would be great. And what we're focusing on is actually reactive oxygen species. So Biologically, the oxygen radicals that are most important are hydrogen peroxide, the superoxide anion, hydroxyl radical, mostly a little less, right? Molecular oxygen's more in respiration. But for signaling, what's important is that reactive, what am I trying to say? The radicals are controlled, right, by electron loss, right? And if there are things that can promote or inhibit this, we can then promote or inhibit, there we go, reactive oxygen species concentrations in the cell and even outside the cell. Another important aspect of signaling downstream of reactive oxygen species, it isn't just a matter of having some or not having some. So ROS, or reactive oxygen species, work on a threshold or a concentration dependent manner where both very low levels and very high levels are unhealthy. You need a certain level of ROS for normal cellular homeostasis to keep things going, and very high levels of ROF actively promote cell death. But in a very not well-defined, quote, moderate range in between, reactive oxygen species, rather than being harmful, are active participants in cell signaling, driving cellular activities like gene expression and proliferation or cell division, new tissue growth. For us in our lab specifically, there is an interesting relationship between a stem cell state and the amount of ROS that it's exposed to, where ROS levels within these moderate signaling levels are able to, at lower levels, stem cells are quiescent, or they just sort of stay stem cells. And as the ROS levels increase, it encourages stem cells to differentiate into other types of cells. And again, too much is how you get rid of old stem cells through cell death. So what, fortunately, I don't have to explain this because of Betony, thank you. But mostly, if radical species are controlling cellular activities, then if we can control the formation of those radicals by changing, say, spin directions up and down, we might have a tool that enables us to manipulate ROS levels and thus stem cell activities in vivo. So not to belabor this, the point of the theoretical model is if you form more radicals, right, with alternating spin states, you actually decrease the amount of ROS that the cell sees. And if you have symmetric spin states, I don't know if that's a word people use, but I just did, it promotes more free radicals, which gives them time to diffuse and increases the level of both ROS in the cell and the amount of signaling that is involved. So because regeneration occurs within a weak magnetic field, so roughly 25 to 65 microtesla, and we know that magnetic fields are able to affect spin states in electrons in other systems, we took the leap and thought maybe we could see if it worked in a whole organism like our worms to control that ROS signaling. So we use a mu metal enclosure that blocks a majority of the magnetic fields, and then we use 2 Helmholtz coils to generate the magnetic field strength that we want. We usually have a control coil in the left that's set at 45 microtesla, which is sort of normal and average. And then our experimental coil on the right, so that we can run experiments in tandem with a mu metal separator so we don't get any bleed through. to study the effects on stem cell activities during regeneration when they're exposed to different field strengths of magnetic fields. And so if we start with that middle fragment, like I showed you before, a trunk fragment, that's going to need to grow a new head and a new tail. We are just going to look at the head part, but what I say also works for the tail as well. We find that at 45, which we're assuming is our control level of magnetic field, since it's an average for Earth normal, and you'll see all of these in the middle, we get what looks to us at least like a normal average amount of new tissue growth at day 3. That's when we're looking at it. So these are the new head tissues at day 3 after amputation. Then we find that the results that we have depend on the field strength. So they vary by field strength. So if we are at 200 microtesla, we can get an inhibition of tissue growth. And conversely, if we're at ranges like 500 microtesla, we can actually increase growth over the amount of normal tissue growth. Now, what we're interested in, of course, is this having an effect on ROS? So we have a general ROS dye here, though lately we have gotten into specific species of ROS, but this one just does general general ROSs. And again, we see that ROSs are upregulated after wounding at the wound site. So again, we're looking at that head wound, but now we're looking at it an hour after amputation. So much earlier than the tissue growth I just showed you. And we see that this response with the reactive oxygen species are known to be required for regeneration to occur. And again, if we look and expose them to 200 microtesla, like we've done on the left, we lose almost all of our reactive oxygen species, at least with the ability of the dye to see it. And conversely, if we look back at our 500 microtesla range, we can increase the amount of ROS that is expressed. And because we are sort of developmental molecular biologists, we that's not good enough. We want to know what's going on in the middle, because there is a big black box between ROS species, even in the cytoplasm, but they might be extracellular, we don't know, and new tissue growth, right? And there's 3 whole days separating them, right? So what we do know from our experience is that activation of certain genes like this heat shot protein 70, which is only important in this context because if the HSP70 isn't expressed, the stem cells in the planarian will not proliferate, and you won't have any tissue growth. And what we found is that just like if you block ROS, where you lose expression of HSP70 and you lose stem cell division, we can repeat that data just by exposing to our levels of magnetic field. So at 200 microtesla, we are reducing the expression of HSP70, and we're greatly reducing the number of stem cell divisions, which you see as those punctate dots in the bottom panels. And then again, conversely, excuse me, at 500 microtesla, we vastly increase the expression of HSP70, and there is a corresponding response in the amount of stem cells, which makes sense since we saw a corresponding increase in the amount of new tissue growth. If we probe a little further in between sort of stem cell division and new growth, what we find is that at 200, we are reducing the amount of stem cells as well as their progeny or their descendants. So these are the cells that are going to go on and differentiate into mature head tissues. And at 500, we are activating both the number of stem cells as well as their progeny. So here we go. Sorry. So what we get is a pathway that functions through ROS signaling to turn on gene expression, to change the cell division of stem cells and their differentiation, leading to the formation of new tissue growth. And some data that I didn't think I'd have time to show, but maybe I would have, is we have now started looking at individual reactive oxygen species dyes, and we find that this does not occur with the weak magnetic fields through what am I trying to say? Through hydrogen peroxide, which is probably the most widely understood ROS signaling molecule, but instead the weak magnetic fields are altering the amounts of superoxide, which suggests that we might be functioning through perioxynitrate signaling, nitrate signaling, and that's a direction we're currently looking in. So signaling, the ROS produces changes in stem cell activities and produces new tissue growth. Weak magnetic field effects can change the amount of ROS that has occurred and thus the downstream signaling, and now those effects vary by field strength. And then weak magnetic fields may excitingly be a new potential tool that we could use to actually manipulate stem cell activities in vivo to either promote it for regenerative therapies or inhibit it in cancerous situations. I just wanted to take a second and talk, because all of that, you'll notice the field strengths that we were looking at were above Earth normal. They weren't very much above Earth normal, right? They're still considered weak magnetic fields, which is generally anything under a millitesla. But once we get outside the Earth, especially in sort of interplanetary space, we're way down to what they refer to as a hypomagnetic field, which I spelled wrong, sorry. What are the consequences? Because we've just seen that you can have vast consequences in cellular activity and new tissue growth and wound healing and that sort of thing by varying the concentration, not the concentration, the field strength. So what you find in the literature is that there definitely are hypomagnetic field effects, right? With a bunch of studies showing that reducing the magnetic field to sub-Earth normal reduces the amount of ROS and thus reduces the number of new cells that are produced. But there are also ones that show that ROS is increased, right, in a hypomagnetic field, and as well will increase the amount of new cells that are produced. So the complications here are varied. There are differences in how we are establishing a hypomagnetic field, whether it's just merely shielding the Earth's normal or compensating by running a field in the opposite direction, or doing what we do is shielding and then applying a field on top of it, which could presumably result in different results. There's also, strangely, no real good definition of what a hypomagnetic field is, with people using terms near zero to mean less than a nanotesla, less than a microtesla. I've even seen it up to less than 15 microtesla, though I think that's kind of pushing it. And as with everything, there is a range of models being used from cells, tissues, and organisms. And the one thing we know about cell signaling in developmental biology is that the same signals can be used very differently in different cells, different tissues, and different organisms at different times, right? So we sort of have to know how the magnetic field effects interact with the known signaling pathways within the cells if we are going to want to use this. So I'd like to thank my lab and my collaborators and the Guy Foundation for inviting me to talk.
Wendy, thank you very much indeed. Very, very interesting indeed. And I'd like to open both Betony and Wendy's presentations to questions.